I hope that we will be moving to the real class hall meetings soon, once the pandemic is over. We are conducting these various topics, topics of interest and the recent research through such kind of meetings, and we want to popularize this as much as we can. Since this topic is going to be the more technical, I see that the DNA sequencing, so much of things, it's specifically meant for a particular kind of audience. That's the reason I see that the attendance is less. Otherwise, no, we used to have 60 to 100 in the Zoom meeting, and once the meeting is over, our YouTube will be there in our Popular Science YouTube channel. So today we have a lecture with the new next generation DNA sequencing technology from Dr. Sushil Chandani. So the speaker will be introduced by Dr. S. Krishnasamy. He is well known to popular science lecture. He was also part of this group, and he was a department. He was heading the department of biotechnology in Madurai Kamaraj University. So it's the time for starting the meeting. This meeting will go for an hour, and after that we will have a question answer session. And I request all the participants to put your mic in mute so that the meeting can go without any hindrance. And we also going to post our link for the feedback. You please fill up your feedback so that we will be keeping in touch with you through the information you are providing, and we will also make use of your feedback to upgrade ourselves. With this introduction, let me hand over the mic to Dr. Krishnasamy to introduce the speaker. Thank you.
So good morning, everybody. It's a pleasure to be at the Popular Science Lecture and opportunity to introduce Dr. Sushil Chandani. Dr. Sushil Chandani's talk on this next generation sequencing is a very apt one given these times. I think we will have the talk in English, a bit more technical, and maybe at the end, if possible, I think somebody may translate. ஆனால் அந்த மரபணு வரிசைப்படுத்தல அந்த மரபணுல இருக்கிற நியூக்ளியோடைட்ஸோட வரிசை இப்போ நம்ம எப்படி இங்கிலீஷ் லாங்குவேஜில் ஆல்ஃபபெட்ஸ் இருக்கிற சென்டென்ஸுக்கு லெட்டர்ஸ் இருக்கிற மாதிரி அதை கண்டுபிடிப்பது என்பது ரொம்ப பழைய ஒரு 50 ஆண்டுகள மேலே இருக்கு. ஆனால் அந்த வரிசைப்படுத்தல அந்த டெக்னாலஜி தொழில்நுட்பம் மேன்மை அடைந்து இப்போ ரொம்ப வேகமாக பண்ணக்கூடிய தன்மை இருக்கிறது. அதுதான் இப்போ நெக்ஸ்ட் ஜெனரேஷன் சீக்வன்சிங். அந்த நெக்ஸ்ட் ஜெனரேஷன் சீக்வன்சிங் இஸ் a technology that has come because of improvements in the sequencing technology, which allowed earlier one DNA sequence determined of one gene from a bacteria, maybe some students' entire thesis and 5-6 years of work. But nowadays, that hardly makes a mention. This is just deposited in the sequence database. And like the next generation sequencing is what is allowing the huge amount of COVID sequences to be generated all over the world. Such a large amount of COVID sequencing technology has been made possible because of next generation sequencing. And I understand Dr. Sushil Chandani will be talking about the most important contribution by Shankar Balasubramanian, his colleague, on the Solexa Illumina sequencing technology. And as he is also aware, subsequent to that, there has been the development of nanopore sequencing from Oxford Nanopore, again involving some people of Indian origin there. So maybe perhaps if he has a time and inclination, Dr. Sushil may also touch upon that, I guess, about how nanopore sequencing is making things much more rapid than the Solexa Illumina alone, which has been there. So Dr. Sushil Chandani is a very apt person to speak on this. He has wide experience, molecular expertise, and has a range of spanned both in time and geographic space, because he started his masters in Indian Agricultural Research Institute, IRA, Delhi, moved on to CCMB, and worked with the renowned Dr. D. Balasubramanian there, and then he completed his PhD in from University of Kalyani, and continued to work in CCMB, and then moved to the LV Prasad Eye Institute for about 4 years, from 96 to 2000. And subsequently, after 2000, for a decade, he was in the Boston University there, working on molecules which are involved in environmental, which are a result of stress, such as NF-kappa B and variety of others. Then he has moved back from Boston in 2010 also, come back to Hyderabad, and been involved in various entrepreneur companies and other industry is there. So right now, I believe he is in the this new biological, the company called the New Biological Entities, and it's a very difficult transition, I know, from academics to go into industry. A lot of effort is needed, and to maintain being in industry, as many of my colleagues who are there in Bangalore and other places can tell, Chandani is well known amongst the industry people there of the next generation sequencing. And I'm sure he has the ability to translate his thoughts into more simple language, as we saw from the article that came in the Hindu, where he introduced by Dr. D. Balasubramanian again. And so, Dr. Chandani, it's a pleasure to introduce you to the Popular Science lectures, and I hope that you will continue to travel with us even after this talk is over, and we can continue to have dialogue and interaction with you over the years to come. So, Dr. Chandani, it's my pleasure to invite you to start the talk.
Thank you very much for your most generous introduction. It's a great pleasure to be with the Tamil Nadu Science Forum, because when I looked at the list of topics which have been covered in your 38 talks so far, I was very impressed with the fact that you have been reaching out to various kinds of subjects and making sure that people who are interested in science will come forward, listen to them, and learn from them. It's a great thing, because I believe that popularizing science is the surest way of expanding the brains of the whole of mankind, actually. And all of us as scientists work towards that end. For example, I conduct science quizzes in schools, just so that children can have fun as well as learn some science and so on. And what your TNSF is doing, I think, is remarkable, and I'm really happy to be here speaking to you all today. I would like to thank my hosts, Dr. S. Vijayan and Dr. Krishnaswamy, for all that has been put into this so far. And I would like to start my lecture now by saying that it will be a freewheeling thing. If anyone has any questions, because some places I have tried to make things simple, but if some places it looks slightly complicated, then that slide only. If you wish to have something clarified, I'll be happy to do it. At the end, of course, I'm open to discussion and so on. That's how it will be. So thank you all, and hope that you all will enjoy this. I'm looking forward to talking to you all. And the obvious place to begin is the pandemic, which we have been going through for more than a year and a half now. And it has awakened us to how important biology, biomedicine, and the allied branches which go with it, ranging from synthetic chemistry to immunology to epidemiology and so on are. And it's amazing how much the lay person, people like my friends who are not at all in science, how much more they know about what happens inside their bodies and so on. Their awareness has gone up a lot. It has not been an easy time for most of us, but yet learning is required because that's how we learn to cope with the adverse conditions. That's how mankind evolved, right? Facing one adversity after the other. So here we are going through another phase in the history of mankind, and hope that all of you and all of us will come out of it without too many blemishes. One thing that has happened during this whole phase is that we have learned a lot about one small little virus which has troubled us so much. And a lot of that we have got to learn about it has come from techniques which have been developed in the not very long past, recent techniques which have evolved to a stage where we are truly glad that we have them with us, because they have made our job of tackling the consequences of this virus much more easier. And next generation sequencing technologies probably stand at the forefront of all these techniques, along with, of course, the slightly older methods of making vaccines and so on and so forth. There will be drugs too, but drugs have a long development time, and I hope that a new class of antiviral drugs will be coming to us in this phase. So I'll start in a very general way by saying what most of you know very well, and that is that DNA and RNA, in the context of life forms, from the simplest virus to us complex human beings, information is stored in the form of DNA, sometimes RNA. And what is the nature of this storage has been a thing which has perplexed human beings for centuries, even millennia. We have said that there is a blueprint to life and we should know what this blueprint is. But for most of the time, our ramblings on this blueprint have been philosophical, metaphysical, even at times, and so on. Only in the last 150 years or so did they start to get a sure scientific footing, starting with work on genetics and so on. You know that there's something which is carrying things across. If you all remember Mendel's experiment, he kind of showed that, yeah, there is a trait and it is inheritable. So what is that trait? And various scientific methods starting from there helped us to finally crack the central dogma of biology, which says that there is this master molecule DNA, which begets RNA and from which we get protein. And proteins are the working entities that we are most familiar with. Information is usually stored in DNA, although sometimes it's stored in RNA. And DNA, RNA even, are usually packaged in the form of chromosomes. Simpler life forms may have just one chromosome, whereas, for example, we have several chromosomes, including uneven X and Y, and several paired chromosomes, and so on. The amount of information is truly mind-boggling. We have 3 billion base pairs of double-stranded DNA in our chromosomes. This is a lot. And the way to study this, I think, for simplicity's sake, the bases of DNA got classified into something like alphabets. And by themselves, they form an alphabet because it seems that life has a language and grammar of its own, which we are still learning. Important trait for DNA is its ability to replicate. In fact, that's the basic definition of life. Usually starts with the fact that you can replicate yourself. You don't need any external agent to create yourself and so on. And so I'll start with the focus on the replication of DNA, because that is where the story of how our interest in sequences and why they are important begins. In the simplest way, the very familiar structure, double helical structure of DNA is presented on the left of the screen. And from there, you'll see that if you want to replicate DNA, you first have to open it up. After you open it up, you start to synthesize the strand. The property here is called complementarity, which is DNA bases are always in complementary pairs. That is, A pairs with T, G pairs with C, and no other way. So if you know one strand, if you can read one strand of the DNA, you automatically know what is on the other strand. And that is the property that is used in replication. That is, replication machinery will just go on adding. It will look at the next base. If that's a T, it will add an A. If that's a C, it will add a G, and so on and so forth. And this way around will slowly build up 2 strands, at the end of which you are left with 2 identical copies of the DNA that you started with. This way, you had a doubling of DNA and consequently doubling of either the cell or the life form in the case of unicellular organisms is achieved. So this is the replication funda we have probably studied from school onwards. I had said that information is stored in DNA, and for human beings, the ability to try to read that information was really been something which was worked hard for many, many years. It started with very simple attempts to kind of chop off one base from the end of a DNA strand at a time and look at it and see, okay, now A has come out, T, a very long and laborious process, which was shortened in the 1970s in the laboratory of Frederick Sanger in the University of Cambridge. Frederick Sanger was one of those remarkable guys who got 2 Nobel Prizes, one for the technology of protein sequencing and the other for the technology of DNA sequencing. So his DNA sequencing technology, although it was also quite laborious, it was chemical in nature. It required, doing lots of chemical reactions. But most importantly, it involved the use of the machinery that is shown in the, is kind of implied in the slide for replicating DNA, which is the DNA polymerase. So the Sanger technique, as it is called, gave us a method of, at a slow pace, at about stretches of 80 nucleotides at a time, you could start to read DNA. Please remember, I told you that the human DNA has 3 billion base pairs. So if you are going to study 80 at a time, it's going to take you a very, very long time to unravel the blueprint of life, if you want to call it that. Somewhere in the process, of course, because of advances in other fields, there was great pressure on, increasing the speed at which you could acquire sequences. Along came the grand announcement that we would do a human genome project. That is, we would have a project which by the year 2000 would have the complete sequences of all the DNA found in human being ready and available to the world. And because of the ambitious nature of this project, many efforts were underway to try to come up with methods of speeding up the process. And a leading method which came along is the work of Shankar Balasubramanian and David Klenerman, also at the Oxford University. In fact, they were working in the same department from which earlier Sanger had done his work of sequencing. Balasubramanian and Klenerman were really not into sequencing. They had both been recruited simultaneously by the department as junior faculty members, and they had interests of their own. Klenerman was more of a synthetic chemist. He was interested in chemistry, and Shankar Balasubramanian had the interest of looking at what is the mechanism by which DNA replication takes place. How does the enzyme, which, the machinery which replicates DNA, does it move? Does it stand at one place and the DNA moves? Those were the kind of biophysical questions that he was intrigued with, and he was trying to work out an answer to these by figuring out a way of keeping DNA motionless and then seeing what will happen to the enzyme which sits on the DNA. Actually, I'll come to that image soon, which sits on the DNA and then moves along it so that DNA replication occurs. So the interest started there. Both of them probably shared very adjacent laboratory and office spaces. So they started interacting with each other, and in the process came up with new ideas. One idea which had come from Balasubramanian was that we should use fluorescently labeled nucleotide. Adding a fluorescent tag to a existing molecule is a method of kind of visualizing it. This has been used as a spectroscopy and in microscopy for many years now. If you want to visualize something, you just put a tag which can be then observed by you. So what they did was they were working on trying to take nucleotides and add a colored molecule. Let's call it a colored tag to it, so that you can quickly say that there are 4 bases. And if I see green color, it will be so and so base. If I see red color, and so on. So that was the chemistry which they were working together on. And the theory that they came up with was that this same method of observing the growth of DNA in the presence of a polymerase could also be used to sequence DNA, because they had observed that they could clearly distinguish that first one color comes, then another color comes, And DNA sequencing is essentially about finding out what base comes first and then what comes next and so on. So they completed their chemistry, and from this, the next generation sequencing method of Solexa was born. I'm not going to go into deep into other next generation techniques now, but parallel to this, there were many, as I said, there were many efforts underway to sequence more and more of the available genetic material on our planet. And in that effort, many endeavors, laboratories, and companies were putting in their efforts. Many of them came out with solutions over time, starting with the Solexa and going on to 454, which later on became Roche, and so on, right up to Oxford Nanopore, which are in the introduction we were already kind of described about. Okay. The company which these two people formed was called Solexa. So originally this was called Solexa Sequencing, but hardly a year after it was formed, the company was acquired by a larger company also interested in DNA sequencing called Illumina. So nowadays, very often you use the term Solexa or Illumina or both of them together. The technology and all that is inherited from the laboratory in which Balasubramanian and Klenerman worked. They also use DNA polymerase to look at how you form a new strand. Okay, and in this slide you can see that you have one existing strand, which I'll call the template strand. I'm sorry. And during replication, there's a second growing strand. And you want to watch what is added to the growing strand at a time. This addition is done, you can see on the image on the right, the enzyme called DNA polymerase, which is a very rapid processor of this particular job of, growing the strand of DNA using the existing strand as a template. In this slide, I have visualized the DNA polymerase, which seems to have something like a clamp with which to hold the DNA. And then it has other portions, which I'm not going to. They are involved in ensuring that it has made a correct decision of adding it does proofreading essentially. As I told you, the language of DNA very often is the language that we use in the publishing industry, in writing, in language and grammar and so on. So it has this enzyme has both a synthetic ability, it's a polymerase, and it also has a proofreading ability. It checks what it has done. Excuse me, I just had a little water. Sorry about that. What Balasubramanian and Klenerman did was they said, okay, instead of just adding any which nucleotide, the usual A, T, G, and C, we'll take those 4, but we'll label them such that the moment they are added, you can actually visualize. You'll see a flash of color, which indicates that a new base has been added, and depending on the color, we will know what base has been added. By keeping record of this, we will be able to come up with a method of sequencing DNA. That was the fundamental idea on which they really capitalized well. And so they came out with the chemistry of it. I'm sorry, which can be kind of presented as schematic in this figure, which I'm showing you, in which you can see that you take a strand of DNA, small piece, not a very large one. In fact, it's an important part of the technology is this fragmentation, which I will come to. And this one you are replicating using this labeled, fluorescently labeled nucleotides so that you get a complementary strand which is colorful essentially. As you can see, depending on the base, it has various colors. And these are the colors which will interpret and get the sequence of what is going to happen. So this is the basic schematic of how next generation sequencing is done. It's called next generation because the earlier methods involved, as I said, very laborious one nucleotide at a time methods, whereas here you are growing the strand and then studying what is happening. So this is a far more rapid method of checking the sequence of a nucleic acid. Very often when I say DNA, it equally applies for RNA too, by the way. So what was the idea which they had? What they did was they would take this large piece of DNA that we have. The chromosome is like huge, okay? Or even if you take small entities, take the COVID virus, it is 30,000 bases in size, and that is not a very small size for handling if you want to do sequencing. It's very difficult to read something for 30,000 without having an error coming up or you making mistakes in recording and so on and so forth. So what they said was, we'll first make small pieces of DNA and then kind of will polish their ends and attach our own adapters to them. Their adapters are also nucleic acids, but it's a sequence which they have chosen, which is unique and is not commonly available in the biological repertoire. And they are using this, attaching these adapters for a particular reason. So what they are getting is pieces which are 100 to 150 base pairs long. This is a very small piece of DNA. And you have thousands of this, even millions of this. If I were to take the human, say, take a human chromosome and then the human nucleus, take all the DNA in the nucleus and break it up and then make pieces of 100 to 150 base pairs, you'll obviously have millions of pieces in your hand. All of them are subjected to the same polishing and a chemical reaction in a test tube, after which you take this sequencing library and you take it to a flow cell, which is actually the heart of this whole process, chemistry of this whole thing. All the chemistry takes place on this flow cell. This flow cell is essentially somewhat like a microscopic slide. It's roughly of that size, except that it's very high tech and it's coated with 8 lanes. And in each of these lanes are small nanovels, each one a few nanometers in size, designed such that it's just enough to fit in a few DNA molecules. It's not designed to fit in like, thousands and thousands, because if you know the size of DNA, you'll know that you need to have nanometer size things to kind of accommodate it. Anything larger than nanometers will be like a small pond as far as DNA is concerned. So the flow cell which they have designed is coated with the single stranded oligonucleotides, which are just kind of tethered. They are just attached to the bottom, and this acts as kind of posts to which the adapters will come and bind. The adapter and the oligonucleotides, which are permanently tethered to the nanovels of the flow cell, are complementary. So they'll go and find each other and quickly bind. So you will end up in a situation where, roughly speaking, each nanovel has one DNA strand, which is a small fragment of DNA sticking out of it. You can see that in the figure at the top right. You can see that it's like, at the bottom you have the tethered oligonucleotide to which the adapter which you have linked to your DNA is going and finding and binding. Now, you start amplifying this. The process called bridge amplification. I will not go into the chemistry of all this. Suffice it for you to know that what you are trying to do is you are taking one single piece of fragment of DNA and making several, up to 1,000 identical copies of that DNA. Okay. And for this, you use a polymerase chain reaction. PCR is something which, again, most of us have learned thoroughly in the last 1.5 years. So that same PCR methods are used to kind of enlarge the number of molecules of that fragment of DNA. You started with one, and you want to end up with a cluster which fits into each of those nanovels, and a cluster which contains about 1,000 clones of the DNA fragment. Why this clustering is required is because when you are doing the chemistry and trying to look for signal of a fluorescent molecule, trying to detect fluorescent signal coming from a single base is simply too much to ask for. The signal would be too faint. So to amplify signal, you're amplifying the copies of the DNA fragments. I hope if there's any queries in this particular part, I will move up to. To this now, designer nucleotides are added. Okay, nucleotides are what I talked about earlier. This is what the chemistry wizards in Cambridge University had come up with. They have two properties. One property is, of course, they are very colorful. And the second property is that after one nucleotide is added, the reaction stops. The polymerase can't go further. It can add only one and then it waits. Okay. So that's the beauty of this technique. What you're doing is starting by taking flow cell, which has got this DNA clusters. To that, you are adding all the nucleotides along with the enzyme DNA polymerase, which will cause the replication of the DNA. Except that the replication happens only for one step, it stops. At this point, you take a camera, a very fancy camera, I must say, and this records an image of the flow cell. That you see at the bottom, spread across in terms of several 9 pictures of particular region as it happens over time. Okay, and each particular place that you stop, you have one particular color on the clusters, depending on which was the last base that was added. If you added a G, you'd get a bright red spot. If you added a C, it would be a green spot. You would be adding a C if the complementary strand, that is your own DNA, had G on, as a 18 with that. Now, the color of each spot tell you what base has been added. At this point, you wash the flow cell, you remove all that is there, that is except the DNA which is tethered to the nanowell, and treat it with a deblocking agent, which now allows it to repeat this. You go through the cycle once again. That is, you throw in some nucleotides, there's designer nucleotides, you put in some polymerase, does its job, adds one base, and after the addition of one base, you again record in your camera what is the color of the most recent addition. This process goes on. Each step takes about 1 second, by the way. It sounds like a lot, but it's quite fast. So, and goes on that way. And in the process, you're collecting the images, from which you can interpret the sequence. I have shown you from the example how we can say that one particular image we are seeing GCTGATC. That's the spot which is at the top, and the second one is the spot at the bottom, which is a long strings of C followed by an A and a C. So that's how we are interpreting. This interpretation is done within the machine which does the sequencing itself. It's a computationally intense job, but it can be done. From here, we come to analysis. So you have taken the raw data, which is the photographic images, and you have converted them into a DNA sequence. I already showed you that. This happens on the instrument. After this comes the real difficult part. You are left with fragments and fragments and fragments of DNA whose sequences you know, and then you want to piece them together to get a single long sequence of a particular chromosome, or more than one. Suppose you have taken 2 chromosomes, you may have to end up with 2 strands of DNA to match and add up. At the same time, you are looking for things like, is this different from what is already known about this organism and so on. You're looking for variants. In the case of the COVID virus, for example, we are all the time worried about, is it a Delta variant, is it a Beta variant, and so on. And that variant is just one difference in that whole region of the spike protein of the COVID. Overall, in the whole molecule of the virus, the RNA of the virus will have maybe 20 or so differences, and you have to spot 20 differences in 30,000. So it's a task which requires quite a bit of effort and so on. But I'll come to that little bit further down the line. So these are the analysis steps you're worrying about. You have to make sure that you are getting the results you want and that they are accurate. Aligning the reads, what I've done is I've taken an example from literature. This is the opening line of Charles Dickens' famous book, The Tale of Two Cities. And what I have done is I've done the fragmenting of DNA, literally, so to speak. I've taken a sentence and fragmented it into pieces. But I have not just taken one sentence. I have taken 2 or 3 copies of the same sentence and 3 copies actually of the sentence and fragmented them. And most of the pieces are with me here. And I'm going to try to piece them together and align them and figure out what is the final long sequence of this sentence. This is the analogy to the sentence that would be DNA. Okay, you can see the pieces can be any way. You'll have a start, you'll have an end. But by the time you have done enough computational work, compared the various parts and so on, you will end up knowing that you are dealing with just one sequence. You are not having more than one DNA now. And it is one strand of DNA, which is, so much long, and it consists of the bases in the following order. You can get them to be arranged in a perfectly fine order. This aligning of the reads of the 100 to 150 base pairs, which you get from the Solexa sequencing machine, is a computationally very intense task, because I told you in the case of a human genome, for example, you will be ending up with billions of pieces, and they all have to be aligned together with no pieces left, free and loose and lying around. Right? It's a daunting task. It takes a lot of time and requires intense computation, but the algorithms for doing it have been slowly worked out, and they are getting better as time goes by. Going back to my schematic, what I had shown you was the first two parts of the screen here. The upper one showing you the fluorescent labeled DNA which you have generated with the polymerase, and then the reading which has been done by the machine, which is the various colors and the corresponding bases. Now you can take this and then align these two pieces together using the commonality that you see towards the right side, and know that, okay, this is the single long contiguous sequence that I'm dealing with these two fragments only. Obviously, in real life, the number of fragments is much, much larger. Now, for cheating, what you can do is you can say, I will take a reference sequence. For example, in the case of the COVID virus, you already know you are doing the, you're looking for this particular virus, okay? And something like it. So when you're putting these things together, when you are doing the piecing together of the various fragments of DNA that you have sequenced, you can use a reference which will, quickly help you to put a lot of the pieces together. And then you'll be left with a small ambiguous ones, which you have to slowly work out in the hope that maybe in that few pieces you might find a variant and so on, because the rest of the molecule is identical. So using a reference sequence actually makes your job easier. And it is needed also because when you take a typical COVID test, when they take the samples from your nose and dip it into that solution, only about 4% of the nucleic acid material there is going to be viral if you are, infected and in prime infectious mode. Okay. All the other DNA will be either the cells which are lining your nasal cavity, or there'll be other bacteria, other viruses, and so on, which are there in abundance. Our body supports a huge ecosystem of microbes, so you will get all of them also. And so when you are doing the sequencing, you have to separate out what you want from all the others. So using a reference sequence will help you greatly in sorting the good from the stuff that you are not interested in. I shouldn't say good and bad here. It's all got to do with what we are interested in. I hope I'm coming through clearly. Can I just ask this question to Dr. Vijayan or
It is fine, actually.
That's fine. I'm not Dr. Vijayan. Call me Vijayan.
Okay.
Okay, that's fine.
Sorry.
So I'm a science activist. I'm organizing the meeting. That's it. So
Yeah,
we get
fine.
Dr. TRG.
Yeah, but the point is, am I coming through clearly or?
Yes. We are able to follow. It's good.
It's coming. Going well.
Yeah.
Thank you very much for that.
We are hearing for the first time many of these things.
Yeah.
Thank you for that.
So it made us easy to understand how these colors are converted into the letters. First, you are converting the colors, then converting the colors to the letters. That's good.
Yeah, that's the basic technique.
Yeah,
It's useful because you have only 4 bases, so 4 colors are easy to do.
Yeah.
If you had 26, like the English language has 26, it can still be done, but more intense technology would be required. But yeah, it's a clever technique, is what I must say. Very.
Now, how come they bring the color? That's the question I have.
Okay. Yeah, so I'll tell you what. For this, you have to go into deep seas and so on. What has been found in many of those, hydra and various jellyfishes and all that live in the deep sea, is that their environment is dark. Okay, so their means of signaling to each other and to attract attention or to catch a prey and all involves using molecules which can glow in the dark. Okay.
Wow.
And that is where human beings got to learn of fluorescence molecules. Okay. And in fact, there was a Nobel Prize awarded for the person who took the fluorescence molecule from one of these deep water organisms, brought it to the laboratory and made it into a probe kind of thing. Okay. So after that, of course, chemists took over. They, they figured out what is the chemistry behind the fluorescence, and they started synthesizing their own variants. That's how you end up with red, yellow, green. In fact, now there are so many colors that you can be happy using them in many ways. There's a whole branch of biophysics called fluorescence spectroscopy and fluorescence microscopy, which uses these fluorescent molecules as probes. For example, if I am looking for, is the antibody that I have made binding to the virus or not? Now, you can do it in a very holistic way, but the simplest way to do it, which you can do almost at the field level, is to take your antibody and attach this fluorescent molecule to it. Okay. So when you put it in, if it binds, you will see it is your virus particle will also start showing green color because the antibody is green, because you have bound the green molecule to it. So this fluorescence, starting from deep sea creatures, has been a great tool for biologists and is used very often. Coming back to my story. So using a reference sequence, as you can see, makes the job easier because you already know. It's like already having read the Charles Dickens book when you were a child. So you remember, starting with it was the best of times and something he said. So it kind of helps you deciding in aligning some portions, after which the rest can be put together. Is the next thing that we have to worry about. When you're taking pieces of DNA, today what happens in today's world, what happens is many of these things, we already know what their sequences are. We may not know exactly, for example, we may not know the exact sequence of one particular species of buffalo's DNA, but we'll know that of the cow, which is very closely related to the buffalo. So we already have reference sequences for many of these things. Okay. Sometimes, of course, you find some totally unknown, but that is getting increasingly rare. So usually it's easy for you to kind of say that this is likely to be so and so and so on. In that context, if you start seeing something different, you have to start worrying it. Is it truly a change that I'm seeing? Is it a variant? Has a mutation occurred? Or is it just something went wrong with the chemistry, some noise? You're dealing with signals of light coming very rapidly, and in the process, something can go wrong. You may catch the wrong color. Errors happen in this whole process. So to do that, you have to come up with methods of ensuring that what you have got is a variant. And this technique, which is largely based on algorithms which kind of keep looking at this, and other techniques which I will come to, which will tell you that you are having a variant or not. The most common type of a variant, the ones we worry about, is called SNP. It's there in the diagram, where just one single base has changed. In this example here on the left at the bottom, you see T has become G. Okay. And T to G change can be very profound by the time you have gone from RNA to protein, because it could be in a crucial part of the protein and you're changing the amino acid and so on. We know, for example, that one variant, which in the case of the COVID virus, which has worried us a lot, is the variant which goes E484K. Okay, from E, which is an acidic amino acid, you have gone to K, which is a basic amino acid. Because of this change, the behavior of the protein and its binding properties and all changed dramatically. But at its root, what has changed is just one single base. 1 out of 30,000 is something that we have to confirm. So variant calling is a very important technique. There are other variations also possible. One common one is called indel, which means insertion or deletion. That is, you have wrongly put in some extra bases, or you have removed some bases from there. Usually the bases are added or removed in lots of 3, because that is how the translation happens. How you go from RNA to protein is that 3 bases of RNA will decide which amino acid comes along. So indels, if they happen, especially in units of 3, can be quite troublesome. You just have missing amino acids and you wonder whether the missing is because of something wrong with you, your machine, or with the sample that you are studying.
can I ask you a small question?
Yes, sir.
What makes some variant is more dangerous than some other variant?
Yeah, that's actually the heart of the whole matter is that when you are looking at the variant, first question is what part of the whole business is it involved in? As I said, ultimately this RNA that we are sequencing is going to become a protein, okay, or most of it is going to become a protein. And in this case, we have that famous example of the spike protein of the coronavirus. The spike protein has two regions, very small portions of the whole molecule, which bind to the receptor of your lung cell. Okay, that is how the virus actually infects you. The virus has to first say that, hey, this is a lung cell, or that nasal cavity lining cells. These are all epithelial cells. Okay, and those cells have a particular receptor on their surface, which actually was designed in evolution for a very different purpose. But this virus, being a parasite, has decided that first I have to find that receptor, because a receptor is something that receives you, as the name suggests. And once you are locked in there, you are likely to enter the cell. Once you are inside the cell, it is up to you to divide and grow and then infect other cells. So you are saying that the virus has evolved a property to bind to the receptor. Now, if you change something in this particular receptor binding region of the spike protein, you may either weaken the binding or you may enhance the binding. Okay. And if you were to weaken the binding, it's actually of great help to us human beings, because any virus which has a weak binding will not infect you and therefore will die out. It has no future. That particular variant has no future. It will disappear. Whereas a variant which binds more tightly than the normal variety, let's call the original virus which came from Wuhan as the normal wild variety. Suppose you have a variant which is binding more tightly, then that particular variant will be more infectious. And more infectious means more trouble for us. And that is what has happened. Every time a variant comes along which is more infectious, it definitely causes more infections to happen. It does not mean that it causes more fatalities. That does not happen, because the two properties are actually not related. This particular variant has only learned how to catch the cell better. It hasn't designed any new machinery to become more fatal and so on. Okay, it's only more infectious. In fact, I would say that the COVID virus is following the general rule book of evolution, which was laid down by Darwin and others after him. And the rule book says that when it comes to parasites, please remember, viruses are parasites. For a parasite, the more the number of people it infects, the better. It should become as infectious as possible. In fact, an ideal virus should infect every available host. By wearing a mask, you are making yourself unavailable. Okay, that is one thing. The second rule is, for a smart parasite, it does not make sense to kill the host. That is not a good thing for the parasite also. So the parasite may become more infectious, but it is not likely to become more fatal. That more fatal might be another parasite which will come along. So the rule book is generally going as per what has been predicted by evolutionists. Okay. So the answer to the question, therefore, is some variants are called by the WHO as variants of concern because they are able to bind more tightly to your lung cell, and in the process are more infectious. I hope
we got the point. Yeah, excellent.
Yeah, so there are the various type of things which can happen. Sometimes you have inversions, that is a piece of DNA literally sits upside down and so on, but they are not very common. Translocation is also something which somehow in evolution it has become quite popular. They are called jumping genes and so on. You see them in, especially in plants, they are very common, but they are also in other places. It's just kind of one piece of DNA for some reason goes and finds another place, and it creates havoc in the whole system. But in the examples that we are studying, the most difficult and of greatest concern to us are the single nucleotide polymorphisms, the one amino acid change or one base substitution which happens, which creates great deal of problem if that variation happens in a critical part of the machinery of the virus. how do you make sure that you have actually found a variant and not looking at something spurious or some error in your sequencing and so on? For this, the term that has been introduced is called deep sequencing. Now, deep is actually, I think, a misnomer, because you're not like digging deep or something like that. What you're doing is you're taking more of the sample, essentially. Okay. So if you were to take the example that I gave you of the Charles Dickens example, in the presentation that I made, what I had done was I had taken 3 copies of the book and taken the opening sentence from 3 copies and cut them up in various fragments. Okay, randomly cut them up into fragments of not very small size, not very big size. So instead of 3, suppose I were to take 30 copies of the book, 30 sentences, and make lots of pieces, and then ask a computer, because 30 sentences broken into something like 200 pieces is too much for the human being to do with his hand. You will give it to a computer, which will be in a position to say, now I am very sure what is the sentence that Charles Dickens wrote, because I have 30 references, 30 different templates to look and match and so on. That is what is done in the case of trying to make sure that a variant is truly a variant and not an error of sequencing. You do this called deep sequencing. It's something sometimes defined as a number. Again, saying, like I said, in the case of this, it's 30. And in the case of the COVID virus, usually the number that's chosen is around 80 or so. That is, instead of just taking a few copies of the viral genome, you take 80 or more copies of the virus and then work with it to make sure that you are actually looking at a variant, a difference of 1 base among 30,000. Okay. So I'm using this book as an analogy. I hope you all don't mind, because it is a sentence. Ultimately, DNA is a long and meaningful sentence, or a book, or whatever you call it. Now, I'll come to the second, which is what are the applications of this technology. I had described in detail the Solexa method. Other methods which have been developed by competing companies also follow very similar kind of principles. In some cases, instead of tethering it to a microscopic slide, as has been done in the case of Illumina, they put them on beads which are magnetic, so you can catch them and so on. So the variations are there, and overall it follows the same drift. Instead of using fluorescent molecules, some people use flashes of light, visible light also. There are some enzymes which are found in the firefly. Those guys are designed to produce light. That is why they are called fireflies. We see them at night. So those firefly molecules have been taken, and the flash of light comes along of different colors, and we know that now A has come, now T has come, and so on and so forth. Okay, so that is another popular way of doing it, and so on. But these are all, I would say, variations on the theme. Ultimately, in most cases, you take DNA, break it up into small fragments, and take each fragment separately and subject it to sequencing. When you're dealing with organisms, you ultimately need to say, instead of comparing the whole genome, I will take something and then see how it is different from one to the other. Okay. It makes sense, because comparing 2 genomes is quite a tedious job and the differences are too many. Whereas if I were to take just one particular entity, then I would be, a more clear ground. As an example, I would say that suppose you were to take human hemoglobin and compare it with the hemoglobin from a bovine hemoglobin molecule or hemoglobin from a horse. They would not be identical, but they would generally follow the same principle, because they are both doing the same job. Okay, because they are both trapping oxygen and carrying it, and they are all using that heme entity, which is why they are called hemoglobin. So if you were to take the protein part of a hemoglobin molecule from different species, you will see that they are largely similar, but they have key differences between each other. So one standard which has been slowly developed in biology is this 16S ribosomal RNA. The ribosome is a key organelle in all living things, and the ribosome has a backbone molecule called the 16S ribosomal RNA. And because ribosome is very important, the ribosomal RNA is generally not been tampered with much during evolution. It has remained, that hasn't changed much. Though, of course, it has slowly evolved and so on. So if I take the ribosomal RNA from one species and then take the ribosomal RNA from another species and then compare the 2 sequences, I can sort of say that these 2 are closely related or they are far away. This is the process by which you kind of develop a tree. Okay, this in biology it's called phylogeny, but ultimately it's a concept of building a tree of life, so to speak. An example of a tree I've put here. This is the dynasty of the Marathas who ruled in parts of what is now Maharashtra. It started with a king, a Bhonsle king, and then his descendants in various branches and what all they went through in life and so on is what. It is a very abbreviated form of the chart. Of course, it doesn't show all these, brothers and sisters and so on. It kind of shows you a general summary, but you are getting the concept of a tree now. I can show relationships between the people at the bottom of the chart, the people who are early 19th century people, and then compare it with the people who, the few people who were their ancestors who lived about 150 years before them, and work out a relationship. Similar thing can be done if I were to take the 16S RNA, which I showed you on the last slide, and then start matching them against each other and so on. And just like the Bhonsle dynasty, I come up with a tree of life, all the various forms of life which are there, and then arrange them based on how closely their 16S RNA sequences resemble. You'll notice that animals is just one single dot at the upper right. The differences between these particular ribosomal RNA in animals is very, very little. Whereas between all the various, between the bacteria especially, it is very, very divergent. The tree is very large. An important part is the tree is ending up with three branches, three main branches. Eukaryotes and prokaryotes is what we learned earlier, but now you say no, it is eukaryotes, bacteria, and archaea. Archaeobacteria being a form which comes between the bacteria and the eukaryotes. So this is the tree of life. And to do this kind of things, you need to do a lot of sequencing. Earlier, most of this work actually began in the older Sanger method of sequencing, but now you can do this in huge quantities and work out very close phylogenetic relationships, as they are called, between various organisms and so on. For example, I'll come to this another application of the NGS, which is the COVID virus and its sequencing. It's amazing that in the last 1.5 years, as of yesterday morning, 2,658,000 viral genomes have been sequenced. It means 2,600,000 individuals were infected with the virus. Samples were collected from them, put into these next generation sequencing machines, and the sequences made available to the public in a database which is called GISAID, GISAID. It's an open database. Anyone can go and look, and it's becoming more and more difficult to handle because the sheer number of sequences is so much. Okay, but it's great. The fact is that it is available, and the fact is that we can today sit and do it in such large quantities. This number.
Can I ask a small question?
Yeah, please.
The number which you are quoting, 2 million.
Yeah.
Does it really reflect the variant from the original one?
No, this is the total number of genomes which were sequenced.
Okay.
That's all. Okay. This is the total number of genomes which were sequenced, and the data has been put in this database fellow is not doing any analysis and so on. The only analysis he's doing is the one that you see in the diagram below, which says which parts of the world have how much of variance. The larger the dot, okay, the circle size, the more the variants there are. And it is actually fairly simple. You can make out that, for example, the small island nation here, which is Fiji, on the right of the screen. It's a small black dot there in the middle of the screen on the right, above Papua New Guinea. It's a small dot because it's a small place and not much chance for variants to spread there as happened. So it has very few variants. Whereas if you take a European country, which is at the top of the screen, okay, France, or you take our own India, for example, in both cases, the dots are large, and therefore it shows that there are more number of variants have been found here. There's no attempt being made to do any kind of judgmental. This is just a database. It does counting and it gives data in a very dry form, but then it has got all the data for someone like you or me to say, I want to go and see what is happening. If you have a large enough computer, you can download all the 2.6 million data, sequences there and do whatever analysis you would like to do on there. It's amazing. This number represents more than 1% of the reported cases. India, I must say, is lagging behind. The sequences in India right now stand at something like not even 0.2% of all the reported cases in India have been sequenced. It's a small number, I must say. The number is high in some countries. Australia, for example, sequences about 60% of all the COVID positive cases. They just go and sequence their DNAs, but then that requires enormous resources. So I suppose they are willing to put in the resources.
Where are the genome sequencing done in India?
Oh, there's plenty of places that you do genome sequencing. It's also available on a commercial basis now. You have labs like Vinta Labs and all. They will, on a contract basis, do sequencing for you. In Hyderabad itself, there is a company called Make My Genome, which will take your DNA from, well, take the cells from inside your cheek, scrape them out, take them, and do a partial sequence of all that, and give it to you on a DVD. Okay, they'll give it to you on a DVD drive. They'll sequence your own genome for you and give it to you. They will not do the complete genome. They are doing only genomes which are, I mean, portions of the genomes which are associated with various diseases and so on. But the availability of these facilities is now commercial. Clinical labs do a lot of these things and so on. And more, the cost is coming down. Cost is something which I'll come to at the end of the talk. You will get an idea of how things are moving now and how much relevant that is to all of us. So this is the first slide to just show you how much sequencing has been done. And then those sequences which you got, you can put them in just the type of phylogenetic tree that I showed you, either for the Bonsney kings or for all the various branches of life, bacteria, mammals, fungi, and so on and so forth. So here, if you do that, you'll find that all the viruses which are broadly called coronaviruses actually fall into two broad groups, which are very dryly named alpha and beta coronaviruses, are coronavirus 2. CoV2 is part of the red set, which is the beta coronavirus. It is not very far from the original coronavirus, the SARS-CoV-1. You'll see that in the list which is given in pink. And that's how these viruses are related to each other. This same data can also be presented in other ways, but this way around, you kind of get an idea of how much is the distance between them and so on and so forth. Another way of showing roughly the same data, though this is a different data set, but you have taken a set of genome sequences and then by doing a phylogenetic tree, place them at distances to show how far apart they are. A bat coronavirus is shown at the very bottom right corner of the screen. It's quite far away. But if you look at the sticks which connect these various dots, various circled virus entities, you'll see that the stick has notches on it, and each notch represents one variant. So between the variant which has infected the yellow is the Chinese one. That is the one which is linked to the bat virus. The one which has infected the bat and this, the difference is not too many variants. There are, I think, about 17 notches or something like that. 17 or 18 notches are there. That is the difference between the, and from that particular one, the original Wuhan virus, I suppose, when you go to clusters A and B, there is more and more variation taking place. 2 variants here, 3 variants there, and they start adding up slowly. So if you were to take any in one corner and match it to another one in another corner, you would be adding up quite a few variants in the process. Okay. The colors correspond to various countries from which these samples have been selected. This is a very early piece of work. I took it because it has only 160 examples. Okay. This was published in April 2020, which means the work was done very early. Today, if you were to make the similar one, it would be simply too complicated. It would look like some network theory diagram or something because you have millions of points. I took this as an example because it illustrates the point I want to make, which is that variation is happening and it is kind of spreading out. It's forming a tree of its own as time goes by. And we look at, just had a question about how many variants. So this is an example of that. This is a study which was done in one hospital in Houston. And what they did was between 2 dates, 2020, somewhere March to February 2021, almost 1 year, all the people who came to that hospital complaining and were tested positive, they sequenced their coronavirus. So one single hospital sequenced 20,453 cases, and they decided to, because they have sequenced them, they started analyzing the variants and so on. Because the variations we learned about actually much later. Back in March 2020, nobody even knew that, we are dealing with variants and so on. So actually variants were already existing then, but they were much smaller in number and so on. And so from this 20,453 cases and genomes which they sequenced, you can see a list which says how many had the various variants that we are hearing so often now. Numbers are quite low. Okay, that's one thing. They're just, like handful, 23 of this, 100. And highest, in fact, is the California variant, which has got 100 and so on. But the important thing is, if you have a data like this, you can use it in various different ways. For example, what these guys did is, because it's a single city and it's a single hospital, they plotted out where are the various variants in terms of the geography of the city. After all, each patient gave them his or her house address and a place of residence. So using that data, they worked out. And what they found was that the California variants, which is on the bottom right corner, B1, 429 and 427, were most distributed in the southwest corner of the city, southwest portion of the city, an area which is called Sugarland. Okay, Sugarland is one of the regions of the city of Houston, and Sugarland happens to be the place which is predominantly Asian. There you go, you'll find Chinese and Indians in great abundance. They move from almost 40% of the population of Sugarland is consisting of ethnic minorities which come from these places. And so for the people who did the study, it was a very clear match. People from China and so on, most of their relatives live in California, because California is where most Chinese came originally. They came by ships and steamers back in the last century. So California is where there's the greatest population of Chinese people. And because of their connections with the other Chinese in the country, people of Sugarland, who are mostly Asian in their ethnicity, are getting the Californian variant. So this is how you use next generation sequencing data to worry about epidemiology, how the disease is spreading, what you can do about it, and so on. Solutions also, any administrator can some way of tackling this problem. I won't go into that because it's a political thing and so on. Do you stop people from traveling, or do you do whatever other measures you can do as administrators? But using NGS sequencing data, you can come up with quite large pictures of how things happen and so on. This was one point I wanted to make about how you can use all this huge number of sequences that are now available. And an example of NGS, this is actually very recent work. And what these guys did was they took all the known varieties of maize. There are 26 varieties of maize which are grown in various parts of the world. Okay, our own variety, I think you won't have any trouble finding out which one is more popular in India. It's very clearly there. But then they took all these 26, did the very deep genome sequencing of all 20 out of them, and then started analyzing the sequences. They found that a large number of the genes were there in all the maize, all the 26 varieties. So fine, those are the maize genome. Let's call it the maize pan genome, they call it. But then other than that, each one has its own variations. And we are talking about a lot of variation, because you'll notice the colors are different, the shape of this thing is different. Lots of things are different about this. Importantly, other differences which matter to people in the fields of agriculture are also there. Some of these are more resistant to pathogens. So what you do is, that this particular variety is more resistant to pathogen. So you start going through all the variations that it has, which the others do not have. Okay. So using that, you can work out a scheme that if I have this particular variation in my maize variety, then I'll develop a maize variety which has got nice golden color, but which is resistant to the pathogen which is bothering us in this particular part of India, for example. So that's how you can use next generation sequencing technology at the genomic level to study and work out some things of benefit to all of us. You can also use it in the context of human health. The thing which is coming up is gene panels. The question that was asked about is commercial gene sequencing available? You can see here. This, of course, is not Indian data. I must say this is a data from US and European laboratories, but all those laboratories are now offering clinical laboratories. They'll just take your sample and they'll tell you whether you have susceptibility to either somatic or tumor cancers or cardiomyopathies, various conditions of the heart, arrhythmias, hearing loss, and so on. Okay, mitochondrial disorders and things like that. For making a sense of any of this, let's take the case of cardiomyopathies, okay, heart attacks. What they say is there are 50 to 70 genes which we know are involved in this thing. And some variations in these genes make people more susceptible to heart attacks than others. So if I am worried about my heart, I am, say, a 55-year-old man, and I'm thinking, should I stop doing this? Should I exercise more or whatever? What I'll do is I'll go to the screening company. They are not going to do my whole genome. They will just look at those 50 to 70 genes. They'll sequence those portions of your genome and then say, yes, you have this variant, so you are likely to be having at greater risk of heart conditions as you grow older. So it's maybe better for you to take advice in terms of your lifestyle, in terms of whether you smoke or whether you drink or whether you watch too much television and eat too much fat or whatever. All those factors you can build in using this knowledge. Okay, so that's how you can use. And therefore, the concept here is not of whole genome sequencing, but just gene panels. You said that for this particular disease, I'm going to use this panel of genes and just study those portions of the genome instead of studying the whole genome, which is a more involved effort. Gene panels is another, and it's coming in very fast. Before you know it, you'll start seeing people talking about now, oh, I have got this particular tendency. It's, you're not saying that there's no 100% here. If you say that you have got two variants in your cardiomyopathy genes, it doesn't mean that you will get a heart attack. It just means that you are going to be more susceptible, and therefore it makes sense to be a little more cautious. This is how we are going to use NGS for monitoring and improving your health. More on that later. In a curious way, it's also been used in what is called paleogenomics. That is, you're doing genomics of extinct species, extinct forms of mankind, and so on, and how that has worked out like. See, the thing with the Solexa method, in fact, with most NGS methods, is that what you need is fragments of DNA. Even if you have a complete DNA, you will first break it up into small fragments, right? So in that context, having a very old DNA is actually advantageous because it's most likely already broken up into small pieces. The disadvantage is that over the course of thousands of years, it has also got mixed with lots of other DNAs, including DNA from modern man and God knows whatever animals were walking around there, whatever plants were growing there, and so on and so forth. So all this together requires more effort in terms of sorting, but having worked that out, it is remarkable to today sit and say that the complete genomes of 3 extinct relations of human beings, the Neanderthal man, the Denisovan man, and the Paleo-Eskimo man, have been sequenced, complete sequence. The DNA has come from various places. It has come from some tooth found somewhere, fragments of bones. Even in one case, they took soil from a cave, and the soil happened to have, people lived in that cave for hundreds of years. So their body, skin would have been shedding cells, whatever, hair, all that was taken as one single sample and subjected to NGS. And slowly you worked out that people lived here, they had these traits, and so on and so forth. Sorry. This information is coming in very useful for us to understand how Homo sapiens evolved out of all this people that came out of Africa and how it happened and so on. And very often, almost all the information is coming from DNA alone because other things just are not there anymore. Proteins degrade very fast, especially in hot conditions and so on, hot weather conditions. So ultimately, you're left with hard samples like bone and tooth, and inside those hard samples, some amount of DNA which has been broken down into small pieces over the years. We now know that it's not just that Neanderthals and humans were two different types of human beings, of hominids, sorry, I shouldn't say human beings. We also know that they interbred. That is, you have genetic component from one side going to the other and so on. Modern man, except for Africans, modern man has about 2% Neanderthal DNA in our genes. Similarly, you have, various percentage of the island of Papua New Guinea in the Pacific Ocean. There you have 7% DNA coming from the Denisova branch of the hominid tree. The tree is shown on the right side, the phylogenetic tree showing how the three different hominids, how they are arranged with relation to each other and so on. Sometimes you can even make out when this happened. You can even say that this particular individual, there's one girl who's been named Denny. She is a Denisovan girl. That's what we know from her DNA. Okay. But her DNA revealed that one of her parents was a Neanderthal and the other was a Denisovan. So we are quite close. Similarly, for human beings, we have documented this thing of, people, Homo sapiens who lived 45,000 years ago, whose genetic component of Neanderthal genetic component was much larger, which means it was more recently had they bred together and so on. This is a very strange science because you are dealing with such fragile things, hardly any piece of information available, samples which are between 45,000 and 100,000 years old, and you can still sequence the DNA, make sense of it, and start making assumptions about what was the color of their eyes, whether their skin was dark or it was fair, all kinds of those things. So this is another application, not application, another very fascinating use of next generation sequencing technology. One big field now is microbiology. And in microbiology, the curious thing is they've come up with something called metagenomics. In metagenomics, what you do is, instead of, normally what you do is you would say this is E. coli or this is salmonella and so on. In a laboratory, in a petri dish, you will grow a single bacterium. And after you have grown it, you will isolate DNA from it and sequence it to give you the sequence of a particular E. coli. In fact, the day the E. coli sequence came out, the complete genome sequence of E. coli came out in the 90s, the whole world of biologists was very happy because finally we had the genome sequence of something that we had studied a lot and so on. It was great, but then it's a very tedious thing. And unfortunately, many microbes do not grow easily in lab conditions. To make them grow is difficult. So how do you do? They grow happily in the sewage and so on and so forth, but it's very difficult to isolate them. So the technique of metagenomics says that you take whatever sample you get. Doesn't matter if it has 10 microbes or 100 different species of bacteria or 2 fungi and 7 bacteria. It doesn't matter. You take whatever you get and you subject it to this next generation sequencing. The algorithms for this are even more involved because you have to separate out one from the other and so on. But in the process, you will see, you take the same methodologies used, you take DNA, you fragment it, you sequence it, and then you start sorting them out. In the process, you may get some which are familiar because you have reference genomes available for them, and some which are relatively unknown to you. And then you can start worrying about what to do with this unknown person. Is it good? Is that bacterium good or is it bad? And so on and so forth. Coming to the cost of all these technologies, the cost of sequencing is like enormously dropped over the years. People who have been using computers over the years know of something called Moore's law. The gentleman Moore was the chairman of Intel back in 1964, and he predicted that computing power is going to double every 2 years. In another way, he said, it also means that computers are going to become half as expensive every 2 years. And now, that is 2019, you can see the line which is drawn in the graph on the lower part of this thing. He's been so far completely prescient. He has made one of the remarkable predictions of how computer costs would come down over the years or computer power increase over the years. The line is more or less straight, okay? And it has been straight because of advancing technologies in how to etch the microprocessors. The lines that are etched on the microprocessor, the distance between them, which was 10 micrometers in 1970, is now 7 nanometers. That is how computing power has gone up. At the same time, costs have come down. What has happened in the case of the cost of sequencing a human genome, as is shown in the blue figure and graph at the top, is that as soon as the popularity of next generation sequencing started, the work that I was describing of Balasubramanian and Klenerman started in the late 90s, and the machines were commercially available by 2005 or so, 2004, 2006. Over that period, the machines started appearing in the market. And soon after that, you'll notice there's a sudden dip in the cost of the sequencing of a genome. Okay. The Moore's law line is there at the top as reference for you to see. The human genome sequencing cost is dropping even faster than, much faster than the cost of the computing power and so on. When the human genome sequencing project was done, it was a large project funded by 7 different countries and all that. It started around 1990 and ended in 2000. Total cost was estimated to be $3 billion, which very fortuitously happened to be roughly the number of bases in the human genome. 3 billion bases, you paid $3 billion, so $1 per base. Today, the cost of sequencing a human genome, a complete human genome of one individual, is just coming below $1,000. And $1,000, at least in the Western brain, is a figure which an individual who is not having a very great, riches and so on, who is not very rich, but is having an ordinary, like, general job, he can think of spending. I'll spend $75,000, 75,000 rupees if I want to know whether I have got this disease, that disease, and so on. So, is mankind ready for something that is being called as personal genomics, is the question. This will be the last part of what I'll describe, what personal genomics can today do for you, and I leave it to you to make a judgment on whether you would say it is worth spending money or not. Okay. So if you had a SD card which contains genomic sequence, then today you'll be able to do the following. On the left side, I'm telling you what you can do. On the right side, what you can learn from those things. One thing is that you can pinpoint inherited disorders. These are disorders where one gene is involved, relatively simple and straightforward. And therefore, things like color blindness, sickle cell anemia, and so on. These you will say, I don't need to do next generation sequencing because very often you know that these traits are there. But then it's good to know that it's caused by what particular genetic defect and so on. Using the sequence, you can also predict what is the likelihood of disorders in which many genes are involved. For this, I had already shown you a figure about gene panels. For example, cardiomyopathy, 50 to 70 genes at least are involved. 50 to 70 genes are known to be involved. They may be more. So, what would this information be useful? You can use it to identify what are the chances of you getting diabetes or cardiac conditions as you grow up. You can use it to identify what are the genetic factors because of which you are having conditions like asthma. You can say that I'm having asthma because of this particular defect in some particular enzyme in your airways and so on. Okay, so you can find out about what is happening based on the predictions about what is the likelihood of this disorders occurring to you. You can also use it to uncover unknown familial risks. For example, in the case of breast cancer, many families in India, at least, its prevalence seems to be increasing is a general notion, but nobody has quite figured out, are we accumulating mutations? What is happening? So those kind of things can be uncovered by you if you had a sequence of your genome and compared it to either a reference genome of people who don't get, or to people or families who have a tendency to get breast cancer. There are some Jewish families, Ashkenazi Jewish families, which are known to have it like it happens. It always happens after the reproductive age of the woman, which means it happens when they are above 50 years old, about 50 years old. So if you can match your DNA with theirs and see similarities in some particular genes, then you need to worry. That's an example of how you can use this for prediction work. You can also use this to kind of tailor-make your own choice of drugs, which will be best for you, and how to do the administration of the drug. Do you take it, what interval, how much can your body tolerate? All these things you can use to minimize the possibility of drug reactions happening with you and so on. After all of us will have some susceptibility to some of the drugs which are made. Nobody really knows. We find out only by trial and error, okay? Other than this, in general, you can say, work out your ancestry. You can figure out who all you are related to. That's something which many companies are doing. It's become a big business nowadays. Huh? We'll tell you whether you are related to George Washington. I'm talking about in the American context. Companies offer that, and people happily pay them money to find out based on some genes variations and so on. Similarly, you can use this information in genetic counseling. So this is what the door is at now. The stage is now with the costs being what they are. Whether it is worth doing yet is a question which is open. I leave it to you all to think for yourself. Thank you.
Yes. Vijay?
Yeah, and thank you. Thank you very much, sir. It was so really eye-opening lectures, and it was very simple, and people like me who don't have any biology connection is able to follow your lecture. It's really great. So I would like to thank you.
Really
so,
nice.
Yeah, it is really it is very interesting lecture, and I see that now this lecture will. So we are going to post it in your YouTube. You'll see that it will hit around 400 very soon. That's what I feel now, because it is easy to follow. Now it is time for doing the question answer. I asked the participant to write your questions in the chat box. If you want to ask directly speak to the speaker, you raise your hands. That we will allow one by one to speak. What I see from the question answer, the first question we got is from Malati Asokan. Her question is, why are the gaps in sequencing DNA?
Okay, why are the gaps in the gaps actually are not real. Okay, there are two ways in which you can end up with gaps. One thing is something called repetitive elements. Okay, now suppose by some quirk a portion of DNA has got just going on for 30 or 35 or 40, sometimes even longer than that. You have just one particular nucleotide repeated over and over again. Huh? These repeating sequences are involved in many diseases and so on. They are also known to be amplified, which means the father will have 35 A's, his son will have 60 A's, his grandson will have 80 A's. It's getting more and more. And the father will die at the age of 58, the son will die at the age of 45, the grandson will die at the age of 26, because the disease has become more and more acute. It is affecting the body more and more. Okay. So, and these sequencing technologies have great deal of difficulty in finding out how many repeats are there. And that is because of the fact that you are making fragments and then you are piecing together the fragments. That is the technique you are using. That technique works very well if it is something like a sentence where each alphabet is likely to be different from the previous one and so on. Whereas if you have, same alphabet repeated for a long time, it will become very difficult to piece it together. So that's why you leave it as gaps in this thing. One is that. The second leaving of gaps happens because of the way we look at sequence data and so on. Suppose I'm comparing two genes, okay? And in one gene, my gene, there is a deletion. I have lost some 10 nucleotides have been just somehow went off during the division, cell division and so on. All right. When before I was born, it happened, and all the cells in my body have that missing piece of 10 nucleotides. When I compare it with the normal person's DNA, what the program will do is it will put 10 gaps, 10 dashes in my DNA because there's nothing there, whereas the other person has got 10 nucleotides normal he is. So that is why you'll observe a gap. So some gaps are definitional, that is, they are put by the fact that you have lost something, and the other gaps are because of errors in sequencing. Because the errors, the people who are doing the sequencing analysis, they realize it, and therefore they just say that from here to here we don't know whether they are 30 A's or 35 A's, we don't know, so we'll leave a gap. I hope that answers your question.
Yes, sir. Thank you so much, sir, for a wonderful lecture. Thank you so much.
Thank you.
Yes, the second question is from Murali. So what is a retest reliability of $1,000 personalized genome? What percentage of base pair out of 3 billion match if that retest is done within a month?
Yeah, so you want to know what is the accuracy?
Please, others, mute yourself, except for the, unless you want to ask the question, ask permission from Vijay. Please, go ahead, sir. Sushil.
Yeah, I'm just searching for the question in the chat. Is it there in the chat?
Yeah.
Basically, I want to know what is the retest reliability of the $1,000 personalized genome, you know? So if I repeat the same test on a blood sample, say 1 month from now, how many of those base pairs will match if I do just a blood sample? That's the first question. The second question is that what is the retest difference if I take if I do this $1,000 personalized genome from a blood sample and the other one being, let's say, tissue from the brain?
Yeah, actually, it's a wonderful question, because the second part especially is a huge field of activity, because we know that each different part of our body has differentiated to some extent. Okay, but we believe that the genetic content is identical. That has been the conventional belief. And at the gross level, it does appear to be so. But is it so at the level of those minor variations? Is a question which would need to be answered deeply. In the context of the $1,000 genome, I must admit, the $1,000 genome is not going to be a very deep sequencing genome. Okay, so what the commercial companies do is they do a complete genome sequence, but all the loci which are known to be related to disease, what we know so far, okay, some 1,200, 1,500 in that range, those loci, they do deep sequencing, whereas the other regions, they do a twice read through. If you do a twice read through, I would say the error would be something like 1 in 7,000 bases, you would have an error, okay. Whereas the parts which are having deep sequencing, you can be more or less certain that it will match. But as I told you, this is kind of introducing a bias in your data. Some part of your genome is not that well sequenced. The other part, which happens to be, of relevance to human health, is more sequenced. That's how, when they are doing it for $1,000, they have to cut some corners, and this is how they do it. Yeah.
Next question. Welcome.
Yeah. Next question is how we can able to understand and how and why mutations happen. Do they occur randomly, or we have observed any patterns? Can you briefly touch upon the CRISPR technology? Sincerest thanks for your great talk.
Yeah, great question. Actually, first thing is, do we need mutations? First question. It's a philosophical question, right? You may say that I am perfect. Why should I mutate? Okay, or any organism is perfect, and so why do I need to mutate at all? When I was showing the polymerase, I showed you that it has got a proofreading mechanism also. The proofreading mechanism, which is part of that enzyme, is designed to catch any errors which is made during the process of DNA replication. Because DNA replication is where mutations happen. Mutations happen where opposite an A, instead of putting T, you go and put one C, for example. Okay. C and T are very similar. They are all shape made, they're similar, they're just different in their charge, density, and so on. So, and once you put a C opposite A, that's it. Permanently, it's going to be there, because next time it replicates, opposite C, it's going to put G, not A. Okay. So you have made a mutation which is going to be inherited by all your progeny forever. Okay. Now, why does it happen? Evolution, you require to have variation. This is something we know from that old Darwinian thing about survival of the fittest. You may be fittest, but your environment is constantly changing. Okay, that is true of Earth all the time. Your environment will keep changing. You will have new conditions, new threats, and so on. And every time such a change happens, only a subset of the original population will be adapted to that particular change. And what is that adaptation? It is due to some mutations. Okay, I'll take the example of the Neanderthal. Okay, all these hominids came out of Africa. They grew in the hottest climate on Earth, and therefore all the ones who were leaving Africa were dark skin, dark hair, dark eyes. Okay, dark eye color. Those three characteristics were there of all the people who came out of Africa. Even today, that is true. We all know that. Okay, but they moved into Europe. They moved because of probably population pressures, or there was not enough food or whatever, or they were just curious and they wanted to explore. Whatever reason, they kept moving. They went into much colder regions. And this happened 200,000 years ago onwards. 200,000 years ago, you went into a cold region. And then 150,000 years later, you met another wave of people coming out of Africa. Neanderthals, they are Homo sapiens. Okay. They have evolved in Africa. They have become more intelligent and they are walking erect and they have whatever, they are wise. Wisdom has come to them. They are also moving out of Africa and you encounter them. What do you see in the person opposite you? The 150,000 years of living in the cold has made the Neanderthal skin slightly lighter. Their hair color is no longer so dark. It has become various shades of brown and so on. Same thing has happened to their eye color. In general, their melanin synthesis, that pigment melanin synthesis has come down because it is not required. Your body has decided, why waste energy making this compound when there is no sun? I don't need protection. So evolution has slowly changed your enzymes so that they make less melanin. So you are becoming to look more lighter in skin and so on and so forth. Now, 50,000 years ago, these two populations met with each other. And we also know they interbred with each other because of genetic and DNA evidence. We know that they interbred. Now what happens is the Homo sapiens who originally started off as black in Africa, by the time they are settling in Europe and intermingling with the Neanderthals, their skin color also started becoming fairer and fairer. Their hair color becoming more and more blonde, eyes becoming blue. So today's human beings who live in the same place where Neanderthals lived also have the same traits. They no longer have black hair, they no longer have black skin, they no longer have black eye color. So that's an example of how variation will happen. You can't help it, but whether the variation is suitable to you or not cannot be said. The big question is are mutations desirable or induced? Whether human beings actually make mutations so that if something happens tomorrow kind of thing. That question is actually difficult to answer. All we know is that in the case of bacteria, it is believed that there are some systems which are error prone. That is, there are some enzymes which replicate DNA under stress and they are error prone. And that error prone means more mutations happen there. Some of the enzymes are so bad that they have 5 mutations per 1,000 base pairs that they synthesize. That is a lot of mutations. Whether that's a selected trait, I still don't know the answer to that. I worked on this particular problem for some time, but answer is not yet clear to us.
Thank you, sir. So the next question I missed to ask, question from Welcome. How to sequence a fragment when there is no reference sequence?
You can sequence the fragment. It's no different. When the sequencing is happening, there is no difference between a fragment and reference. And doesn't matter. It's just a piece of DNA. What will happen is you'll have to be a little more careful in saying that this is the sequence of this fragment. Okay. As I said, the simplest way to do it is to sequence it more than once. You sequence it 10 times, you'll become a little more confident. 20 times, you're even more confident and so on. That's what you can do. Otherwise, in terms of the machine, it makes no difference to the machine. It's only to us who want data quickly. We want to finish the job quickly. We want to know whether, delta variant is come or it is the other variant. So those questions we want to answer, you only kind of go with that particular wisdom and use a reference gene. Otherwise, it's fine. You discover unknown microbes very often are discovered purely by sequencing. Nobody has seen that microbe. Nobody knows what it does, but we know it is there because its DNA sequence is there. Complete genome sequence is there. The organism is even given a name, but then that's all. We have never, no human being has consciously studied that particular microbe. There are many examples of such things. Yeah.
Another question from Benjamin Prithviraj Salaman. So what are the exactly external factors can affect human genomes in terms of evolution? For example, hereditary diseases and so on. Is there any technology that we do to predict that?
Predict that. And prediction is actually difficult. But why do we have hereditary disease is a problem which has vexed mankind. The greatest example is the sickle cell anemia. Okay. The sickle cell anemia is caused by a single mutation, one amino acid change just like your spike protein delta variant. It's a one amino acid change in the globin part of the hemoglobin molecule. Okay. That's all that has happened. And what is the result of that is the oxygen carrying capability of that particular variant of hemoglobin is different from the normal, slightly. But for some strange reason, the red blood cells which have this hemoglobin are no longer in the shape of a disc. Our normal red blood cells are like coin shaped. Okay, they are discs, like discs. Whereas the red blood cells for people who have this particular mutation are the shape of a sickle. Now, it's definitely a deleterious trait, which should not have been selected in evolution, right? Because your oxygen capacity is less. You actually have less energy as compared to the normal person. So why did it get selected? It got selected in some parts of Western Africa, Nigeria, and so on and so forth. And these parts of Africa happen to be the places where malaria is endemic. Okay. Malaria is endemic in these places to such an extent that almost everybody is having malaria all the time. That is throughout their lives almost. Okay. Not continuously, but it will keep coming yearly, once in 2 years, and so on. It's considered just normal part of their lives. The parasite which causes malaria, Plasmodium, its life cycle is completed in the red blood cell of human beings. Okay. That particular parasite cannot infect the person with sickle cell anemia because the sickle cell anemia's red blood cells is shrunk in size. It becomes a shape of a sickle rather than a disc. Okay. Which means a person who has sickle cell anemia won't get malaria, or at least is unlikely to get serious malaria enough to cause him harm in health and so on. So sickle cell anemia has got selected there because it's an advantage actually, even though it's a disadvantage in the normal sense, it has been an advantage in that environmental context. So most mutations are selected in the environmental context only. If the environment changes, we'll have some other mutations taking over and so on. So I hope that answers your question, Benjamin.
Yeah, that's the last question is, can you briefly explain DNA fingerprinting in human in forensic? Thank you so much for the wonderful session.
DNA fingerprinting is essentially the technique that was superseded by next genome and next generation sequencing. Okay. In DNA fingerprinting, what you're doing is you're saying that if I take an individual's total DNA, then it has regions which are completely identical in all cases, and some regions which are different from the others in terms of single substitutions. You don't know what those substitutes, you don't know whether it's in an important protein, all those things we are not worried about. All we know is that the DNA has some variation in this portion of the chromosome, in this portion of the chromosome. So you work out like a battery of changes which are there in a susceptible regions of DNA, let's call it. Which means if you take 10 people, for them, those 10 will be different. And the chances of, suppose I have chosen a battery of 150 variable regions of DNA, 150 regions which are susceptible to this polymorphisms as they are called. The chances of all 10 having the same 150 is close to 0, okay? Out of 150, at least one will be different between me and the guy next to me. Same is true of the others also. Therefore, it becomes like a way of identifying an individual. How would you identify an individual? You would identify him by saying that I'm going to take your small amount of DNA. You hardly need any DNA from it. Just digest it using some particular technique and run it on there's a gel for running it, which quickly will tell you which are the differences between you and the other person. There are even more simplified methods of DNA fingerprinting, which can be done in the field. Literally, you can do it outdoors. You don't need to have large machinery, electricity, nothing is required. Battery operated, you can do off and so on. And so it's of use in various ways and so on. For example, the Indian Army, every soldier has got his DNA typed. Your DNA typing is done. So if everyone knows your signature kind of. So suppose a soldier were to die under some harsh battle conditions to a point where his body is not identifiable, maybe a bomb or whatever, then DNA fingerprinting will be used to figure out who he was, and he'll be given a proper and dignified burial. So that's how you use DNA fingerprinting for identifying. The cases which you hear most about DNA fingerprinting have to do with things like paternity and so on. There, what happens is the same thing. You take two individuals, the chances that their child will have to have the variations which you have, or a mix of the variations that these two people have. So if there is a question of who is the paternity question comes into play, then if you do the DNA fingerprinting, if the fingerprints of the father and mother and child all match, then you are confident that it's no problem in terms of paternity. If they don't match, then you can say the paternity is questionable and things like that. So that those are the cases we hear most about DNA fingerprinting. What I must say is that as the cost of next generation genome sequencing comes down, you are going to have less use for DNA fingerprinting, even though it will not be replaced because it's very handy, it's very rapid, and so can be done. Yeah.
Thank you, sir. So all questions are over. So before closing it, I have one simple question. So this the next generation sequencing has revolutionized the identifying the signature of individuals now.
Yeah.
So having such a wonderful thing in our hand, how come still the racial supremacy or the division of people on the basis of religion, caste, everything is still going on? So how can we address this question? Of course, it's something related with society.
Yeah.
So
it's a sociological question which has deep bearings in genetics. Okay.
Yeah.
I'll give an example. This was a study which has not been quoted much. In fact, people will hate me for giving this example. But in the US, because a lot of genome sequencing is going on, and they have got laws which kind of ensure that the identity of the individual is not revealed. Okay. That your genome sequence may be available in the public domain, but your identity is not revealed. But there are ways and means, essentially hacking methods by which you can kind of connect that, oh, this genome sequencing was done in this particular, somehow they figure out whose genome that is, which is not good for privacy points of view and so on and so forth. And also there are some people who openly admit that, yeah, this is my genome. They put it in the database, so-and-so's genome, doesn't matter. If you find that I have got some disease condition, fine, doesn't matter to me, they put it there. Anyway, people sifting through this data find one thing. They find that in places, in populations where there is greater degree of racial supremacy, I'm talking about the US context, where racial supremacy is mostly got to do with white versus black. Okay. What they find is, very often, if you look at the genetic traces of these people, you will find that the person who is white actually has a small degree of black and so on. This is what people say, like, okay, this is hearsay. And from that, you say that, oh, that man is racist because he's more racist than the others because he is actually partly black. Who knows? I mean, so they are the questions which you can address using next generation sequencing, maybe. But then these are all things which are there. I hope you're getting the point I'm making, that some elements of what we call as biases in human beings may be based on what is in your genes, but in my opinion, most of it is there in your mind. It's all got to do your mind and nothing else. So it's a sociological question which is going to be very hard to answer beyond this.
It's purely on political mobilization purpose. That's what it's going on.
Makes sense. The sense that ultimately those are also traits, and we believe that any of the traits that we human beings have some kind of genetic basis, right? So if you say that, then maybe there is something here too. Answer is not no. It's all. Yeah.
Thank you.
So,
Vijay?
yeah, we can close the session. So actually, it was a wonderful and nice session for 2 hours, and I feel that the audience are from a specific field, so they gained a lot from Dr. Sushil, and I should thank Dr. Sushil because he has immediately accepted when I sent a message to him saying that we want to have a popular science lecture, and he has accepted that, and he has given a wonderful lecture. I think we will have a future lectures as well with with him because he is explaining. His lecture is so popular that everyone is able to understand. People who do not have any connection with biology is able to understand what he is saying. It's really a wonderful lecture. I thank you very much, sir. And I also thank all the participants, who are patiently waiting to complete the session. See, many of us has left after once the lecture is over, but still people are there to listen what it has been transpired in the question answer session. I thank all of those who has attended the lecture. Thank you very much, and I thank Dr. TRG, who is guiding us to have such a wonderful lectures. Without TRG we can't have 38 such lectures. And I thank my colleague, who is doing the background technical work, Mr. Sundar, who is here. And thank you very much, and thank you all. Thank you very much, sir.
Thank you very much, sir. First, a wonderful audience and such nice questions. Thank you very much.
Thank you.